In our bodies, before mRNA is used as instructions to make proteins, it can be cut into smaller sections and rearranged in a process called splicing. During splicing, protein-coding regions of mRNA called exons are kept, and non-coding regions called introns are cut out and removed. mRNA splicing is an important step in the transcription process, because without removing the introns, the correct proteins cannot be formed. And if something goes wrong in splicing, disease could even form. For splicing to run smoothly and successfully, a large complex called the spliceosome has to be involved to act as a pair of scissors to splice away the introns. And during the assembly of this complex, a very important splicing factor called SR protein comes in to help, which is what we're going to focus on in this video. SR proteins are named because they contain a protein domain called the RS domain with long repeats of serine and arginine amino acid residues, whose standard abbreviations are S and R respectively. SR proteins are not only important for spliceosome assembly, but also critical for splice site recognition. That's why if we take a closer look into the protein again, it contains at least one RNA recognition motif. For SR proteins to function properly in splicing, phosphorylation of the long repeats of serine and arginine residues is needed. This is because phosphorylation mainly plays a role in the regulation of the localization of SR proteins. Different states of phosphorylation can direct the SR proteins to different locations in the cell. But what does that mean? Let's take one of the fundamental SR proteins, serine and arginine-rich splicing factor 1, as an example. The protein responsible for the phosphorylation of SRSF1 is SR protein kinase 1. And for SRSF1, there are two states of phosphorylation: hypophosphorylation and hyperphosphorylation. Hypophosphorylation means around 10 to 12 serine-arginine dipeptide sites are phosphorylated, and hyperphosphorylation means around 20 serine-arginine dipeptide sites are phosphorylated. Hypophosphorylation causes SRSF1 to move from the cytoplasm to the nucleus, while hyperphosphorylation moves it from nuclear speckles to the splice site for splicing. But something about these phosphorylation events started to worry scientists when they found out that the dysregulation of the events is linked to cancer. They found out that when SRPK1 is overexpressed, or SRSF1 is phosphorylated all the time, a vascular endothelial growth factor, which promotes the growth of new blood vessels, called VEGF-A, will be spliced into one of its isoforms, VEGF-165, a pro-angiogenic gene causing more blood vessels to form and tumors to grow subsequently. And since the dysregulation of these phosphorylation events is tightly linked to a number of cancers, SRPKs have been considered as potential therapeutic targets. In fact, one of the professors of the School of Life Sciences in our university, Professor Ngo, is currently working on a research project that focuses on developing inhibitors that block the interaction between SRPK1 and its substrate SRSF1 during phosphorylation to combat cancer. By studying the structure function of SRPKs and their substrates, currently Professor Ngo and his team has designed a protein-protein interaction inhibitor of SRPK1, docking blocker of SRPK1, DBS1, that specifically blocks a conserved substrate docking groove unique to SRPKs. Since DBS1 is rich in arginine, not only can it effectively compete with the RS domain of SRSF1 at the SRPK1 docking groove, but it would also allow DBS1 to enter the cell more easily. As DBS1 can effectively inhibit the binding and phosphorylation of SRSF1, VEGF-A would be spliced into the isoform VEGF-165B, an anti-angiogenic gene, instead of VEGF-165, a pro-angiogenic gene, and eventually inhibit angiogenesis and tumor growth in vivo. Apart from SR proteins, hepatitis B virus core proteins are also substrates of SRPK1. That means aside from cancer, the inhibitor may also help combat hepatitis B, a viral infection that attacks the liver. Hopefully, in the future, the stability and delivery of this inhibitor could be enhanced and developed into a potent drug candidate.